Ladies and gentlemen, it's a great honor to be chairing the first session with Professor Ho Kim. Professor Ho Kim is the Dean of School Public Health, Seoul National University. I am from the National Institute of Environmental Health Sciences from National Health Research Institutes of Taiwan. And it's with our great honor and pleasure to introduce our first speaker, Professor Brenda Eskenazi. Brenda is a chair professor of the Maternal and Child Health and Epidemiology, the School of Public Health, University of California, Berkeley. And she is currently the director of the Center for Environmental Research and Child Health, a National Advisory Environmental Health Science Council, chair of the Neurodevelopment Working Group, NIH Office of the Directors, and many other roles. And I have the great honor to be seeing her in the Hanoi, I believe. She was an advisor to the arguments between the US and Vietnam for Agent Orange. So I think she has a lot of experience in environmental epidemiology. Let's welcome Professor Eskenazi.
Good morning, and thank you for the invitation here. And thank you for the introductions. So today I'm going to talk about environmental factors affecting the health of our children in the 21st century, what I see as some of the main issues. So many of us do children's environmental health research, and the reason why we do it, the reason why we're interested, is because we think that fetuses and children are more susceptible. We all know now that many chemicals pass through the placenta and the blood-brain barrier, and that children are different than adults in many different ways. And we think that they're most susceptible. So for the last almost 20 years, we've been working on a study called the CHAMACOS study, which stands for the Center for the Health Assessment of Mothers and Children of Salinas. It's a longitudinal birth cohort study investigating the health effects of environmental exposures in low-income Mexican-American children living in the Salinas Valley in California. The Salinas Valley is considered to be the salad bowl of the United States, and this is what it looks like. In the years that we started the study, which was 1999, there was about 500,000 pounds of organophosphates sprayed in the Salinas Valley. In 1999-2000, we enrolled 600 pregnant women, and these were not the usual birth cohort study in the sense that they were probably the hardest population that I could find to follow. They were almost all Spanish-speaking. They were born in Mexico for the most part. They had been in the United States for less than 5 years. We now know that probably around 75% were not, we call it undocumented, but you might think of it as illegally in the United States. They were not well educated. About half of the women worked in agriculture, and if they didn't themselves, they lived with people who are agricultural workers. Our children are now 18 years old. We have followed them many, many times over the last 18 years, and you can see all the time points here. We have seen them from almost being fetuses. We didn't look at the ultrasounds, but to now being adults. They're entering adulthood, and that actually poses a lot of different issues in terms of now they're fully consenting themselves. We collected urine samples during pregnancy, and we measured in those urine samples dialkyl phosphate metabolites, which are non-specific measures of organophosphates in the urine. And the National Health and Nutrition Examination Survey, which is a general population survey of the United States, was at the very same time also measuring these same DAPs in the urine of the US population using the same methods that we did because we used the same laboratory. And you can see in the blue that the distribution of the organophosphate metabolites, the DAPs, were much higher in CHAMACOS than in the US population. And that stands to reason given that many of the women lived in agricultural, all the women lived in an agricultural area, and half of them themselves were farm workers. So over the years, I'm not going to go through every single study, but I'm going to summarize it on this slide and then move on. The organophosphate metabolites, or those DAPs, during pregnancy were associated with shortened gestation, abnormal neonatal reflexes, decreased mental development in toddlers, poorer verbal abilities in preschool, pervasive developmental disorder at 2 years, attention problems in school age, lowered IQ at age 7, poor executive function at 9 and 12 years, and lower scores on social responsiveness scale, which is an indicator of autism trait, at age 14 years. So this slide is only around neurodevelopment. We have also looked at a lot of other health endpoints that I'm not going to go into today, but I wanted to bring this up and emphasize the neurodevelopmental aspect of my talk as a case in point. We have also published that there may be a subpopulation that are more susceptible to organophosphates based on the ability of their mother to metabolize organophosphates during pregnancy and based on genetic predisposition. And we were particularly interested in the PON1 gene because it helps to encode the enzymes that detoxify organophosphates in the body. It also does a number of other things around oxidative stress. We found and we've published that the level and efficiency of the enzymes that are encoded by this gene, depend on your genetics, which whether you're homozygous or heterozygous, and also your age. So you can see on this slide, this part of the slide at the bottom, that here are the levels of enzyme, and this is enzyme activity, in the mother as an adult. And if you look at 6 months, 2 years, and 7 years in the child, it's not until about 7 years of age that we see adult level of enzyme activity. This clearly suggests that children do not have the ability to detoxify organophosphates to the same capacity as adults until they reach 7 or 8 years of age. So it means that they're going to be more susceptible to the effects of organophosphates. We have actually mapped the gene. So that means that if Ruth and I are exposed to the same level of organophosphates, but I have a different genetic makeup than you do, that I might be more affected by that exposure. And so we said, what if the mothers had lower level of enzyme, meaning they had lower ability to detoxify organophosphates? What is the relationship then of organophosphates and, let's say, child IQ in those women that had low levels of enzyme versus moderate levels of enzyme versus high levels of enzyme, meaning that they could really detoxify the organophosphates? So we actually looked at that. We looked at the full-scale IQ, and the standard deviation is 15, and the mean is 100 in an IQ test. And so if the mother had low levels of enzyme, there was a 10-point on average drop for each 10-fold increase in those metabolites during pregnancy, the urinary metabolites that I said I measured. But if the mother had a moderate level of enzyme, then the child only had a 5-point drop on average. And if the mother had high levels of enzyme, meaning she could really metabolize those organophosphates, the child was relatively unaffected. So it's not just the exposure, but it's also one's genetic makeup. Over the years, our research and research of others in California has contributed to the decline in agricultural use of organophosphates. So if you look at this slide, this is something called pesticide use reporting data, which is unique in California. And in the red are the use of organophosphates in millions of pounds over the course of the last more than 2 decades. And you can see that organophosphates have sharply dropped. Here's when the CHAMACOS study began in 1999. Sorry, I have to keep switching these. Our study has not just focused on organophosphates. We've looked at many other chemicals, lots of other chemicals, flame retardants. We've shown relationships with neurodevelopment and other endpoints, thyroid hormone. We've looked at personal care products, phthalates, phenols, parabens. We've also looked at BPA. And we have been concerned because just as we finish doing our research, something else comes out. And we don't necessarily even know how to measure that's something else in the human body. We don't even know what that something else is, but we know that there's something else being used perhaps instead of BPA. So for example, BPA has been substituted with structurally related compounds. This is a list of them. And we have little research on those substitutes. So one of the concerns that I have about children in the 21st century is that we're what I call just whacking the mole. This is a game that kids play where you just push one little mole down and up pops another mole. And that's really what we're doing in children's environmental health. We go down with one and up pop the regrettable substitutes, and we're always lagging behind in our research, trying to understand what's going to be coming out and what those effects are. And by the time we do our research, something else pops up. And that's one of my greatest concerns about children in the 21st century. We don't know that proprietary information. We don't know those chemicals. And we know that many of them are going to be toxicologically similar to the ones that we already found research showing associations. So here's a case in point, going back to pesticides. So if you look at the same slide that I showed a second ago, and you look at the organophosphates, yes, they're decreasing, but in California at the very same time, look at this green line here, there has been over the last couple of decades a steady increase in the neonicotinoids. Those are the same chemicals that are been considered responsible for the bee colony collapse. And we know virtually nothing about the effects of neonics on children's environmental health. At the very same time, we see, a steady increase in glyphosate. Again, as other things are decreasing, other things are increasing, and it's only very, very recently that we can even measure neonics and glyphosate in the human body. So there's been virtually no research on these chemicals and the health consequences. So there are many chemicals in the mixtures that we're exposed to. Our studies, and the studies of probably almost everybody sitting in this room that does research on chemicals in human health, is that we look at one chemical or one class of chemicals at a time, and that's not what humans are exposed to. We're exposed to swimming pools, mixtures of many, many different chemicals, and we've only just begun to think about how to look at those mixtures. So if they're toxicologically related, like for example, dioxins and furans, we can add them up and get a TEQ. Where if we're looking at organophosphates, maybe you can look at a depression of acetylcholinesterase, relative differences, but for the most part, we don't know how to toxicologically put different chemicals together. So one of the other concerns I have about children in the 21st century is that we really need to think about how we're exposed to increasing compounds and increasing mixtures that we know nothing about. Wrong one. So fortunately, as I said earlier, in California, we have something called the pesticide use reporting data, which allows us to look at the amount of pounds of all the different pesticides that are used in agriculture. And so you can see at the top, about 500,000 pounds of organophosphates were used, but about 3 million pounds of fumigants were used, and almost 500,000 of the manganese-containing fungicides were used. And so almost all of these pesticides are neurotoxicants. So how do we put them all together if they have possibly different mechanisms of action? And what do we do with the fact that right now, what we typically do is if we're very sophisticated, which is not very sophisticated at all, is that we control for another chemical when we're looking at another chemical. And that isn't a very good way of doing it. And sometimes if we're super sophisticated, we look at interaction, but that's assuming a multiplicative interaction. So again, not very sophisticated. So what do we do? And so many classes do have similar associations with neurodevelopment, maybe through different mechanisms. We also have a problem because many of the different pesticides don't even if we look at organophosphates, they don't devolve to the DAPs. So about 20% of organophosphates aren't even picked up in the DAPs that I measured. And as I said earlier with the neonics and the glyphosate, they've just be able to be measured, but another pesticide like acephate, which is widely used, doesn't even have a biomarker. So we're dealing with chemicals that don't have biomarkers, we don't know how to measure them, we don't know how they operate, and yet we want to be able to look at them as mixtures. So again, we have this pesticide use reporting data, and it's mandatory in California, it's enforced, and there's no evidence that it's underreported. It provides the active ingredient, the pounds applied, the application date, the GPS coordinates of that application, and the crop treated. So if you look at on this correlation matrix, you see that here are a bunch of different pesticides. Some of them are organophosphates, but there are other classes like pyrethroids and nicotinoids over here. And here's like a correlation matrix. The more positively correlated are in blue. And you see that many of these pesticides are intercorrelated. And that would make sense because if you're spraying lettuce, you don't usually spray with one pesticide, you spray with multiple different pesticides. So they're highly correlated. And that makes it more difficult to be able to look at them because they are correlated. So you can't exactly put them all into the same multivariate model because they'll cancel each other out. So in 2015, there was a meeting at NIH, a very important meeting, which said, hey, listen, we've got a problem here. We've been looking at one chemical at a time. Let's look at mixtures. And they said, we need to come up with statistical ways of dealing with this. I'm not a statistician, but I'm going to try to do this. And they said that there was instability in the effect estimates by putting all of these variables into the same model, because that was not going to work. And there was problems with inference, and you need very large sample sizes because there's loss of statistical power when you put large numbers of covariates together. So how do we deal with it? And so one way that was suggested was using applied Bayesian profile regression to investigate the joint use of different chemicals. And we decided to apply that to the joint use of pesticides and look at it in relationship to that full-scale IQ that I measured earlier. And we use the Bayesian methods because different pesticides are very highly correlated, like I showed earlier. And what we did is we looked at how these pesticides clustered on the effect of IQ. So in other words, IQ at 7 years was the dependent variable, and then we looked at the clusters in the Bayesian model. And what you see here is that two different clusters showed to be most related to the full-scale IQ. Cluster 1 and cluster 7, you can see that on the side there. So then you can then look at all of the different pesticides here along the bottom, and you can see what makes up cluster 1 and cluster 7. And we looked at each of the pesticides based on the PUR data by quartiles. And so you can see that cluster 1 is driven by actually high levels, the highest quartile, fourth quartile, of every single pesticide that we looked at. And cluster 7 is driven particularly from a carbamate and acephate. Now remind you, acephate can't even be measured in the human body. So here we see that two particular clusters seem to be most related to IQ. Now, the reason why I raise this is because as we enter the 21st century more and more, we're going to have to be dealing with mixtures, and we're going to need to be more sophisticated toxicologically and statistically, and this is just one method that we use to be able to look at the PUR data, the pesticide use reporting data, and look to see what's driving that association in IQ that we initially saw. So that's another thing that we need to think about in the 21st century. We have so much more to do. There are 80,000 chemicals that are out there now, and this is only going to increase with time, and only a few have been studied to determine the potential effects on children's health. And looking at mixtures of chemicals requires larger sample sizes to be able to do it. Fortunately, in the United States, we just started the ECHO Consortium, which is a consortium of cohorts around the United States so that we can begin to look at, in an N of about 50,000, the relationship of different chemicals and children's environmental health. And 50,000 is only half of what is happening in Japan or China or Korea and other countries in Asia that are going to be able to amass huge sample sizes to be able to look at mixtures in the 21st century. So I've talked about some of the factors that affect children's health, in particular genes and pesticides. And as you can see on my figure here, there are many, many other factors that affect children's neurodevelopment, in particular, as my case an example here. Many of things in the larger environment that have to do with social factors, many things in the immediate environment, and also the host factors. And we try to control for those covariates in our statistical models. A very important document was written in 2000 by the US Institute of Medicine, now with a different name, called From Neurons to Neighborhoods. And in that document, it said that the effects of early life adversity was key to looking at the development of children. They outlined a number of domains of social adversity, from learning environment, maternal adversity, family dysfunction, stressful life events, economic adversity, and maternal and child interaction. And those of you sitting at ISEE are saying, what does this have to do with us? This is not the environment we study. And I'm saying as a neuropsychologist that you can't separate these things, because in Salinas, not only do they have high levels of pesticides exposure, but the population is almost all immigrant. About a third of the population live at the poverty level, and those that don't live at the poverty level live within 200% of poverty, which means they get federal aid if their children are citizens. There's 2.5-fold the homicide rate in the US average. We cannot drive a blue car or a red car into the community because of the gang activity in that area. 10% of the high school students report being in gangs, and that's a way underestimate. And Salinas is one of the 6 highest communities nationally for youth gang violence. So if I just look at pesticides and neurodevelopment, I think I'm missing a large portion of the picture here. The adversity in a CHAMACOS cohort alone, you can see high food insecurity, living at poverty, maternal depression's high, stressful life events is high. These are kids that are undergoing many stressful life events in addition to pesticide exposure. So I don't know how much this is making the news in Asia, but it's very big in California. Right now, we are going through immigration reform, or not reform, as the case may be, because families are being separated. This is a very live issue in my population. About 75%, as I said, of the mothers are probably not legally in the United States. I hate to use that word legal, but they're undocumented. And the kids are all US born, so they're all American citizens. So we're talking about the impact on American citizens about the fact that their mothers may be deported. And so we decided to actually look at that in the CHAMACOS cohort. And as you can see, we did a deportation scale. Between a third and a half of the children, the US citizen children, are fearing the deportation of a family member. We have now begun to look at the effects on the health of the children. They are undergoing incredible levels of anxiety, sleep deprivation. The mothers are showing cardiovascular problems. And so we know that all of these stressors affect not only the central nervous system, but the autonomic nervous system. So we said, okay, let's begin to look at how these factors affect the autonomic nervous system. And so you can look at heart rate. We look at it in a more complex way that I'm not going to get into. And we have shown that children in the CHAMACOS cohort of lower social class, and remember, all these kids are of low social class, so it's lower than the others that are already low. And we see that there is a difference in the amount of autonomic nervous system activation over time as measured by the heart rate, that those kids that are of low social class have higher heart rate at rest, meaning they're already activated because of living in poverty and living in adverse situation. We also have shown that living in adverse conditions affects actually neurodevelopment, and this is controlling for a lot of covariates that I didn't write down, which I should have. At 9 years, remember we showed that organophosphates are related to executive function at 9 years, but so is social adversity related to executive function at 9 years. And we've published a study looking at IQ, where we looked at all the different adversities and came up with a scale and showed that those children that were exposed to a high level of maternal adversity and child adversity during the childhood, and also exposed in utero to higher levels of those OPs as measured by the maternal DAPs, were the ones that were most affected in terms of their IQ. So we're seeing an interaction of the two. If you want to look at DNA methylation, we've looked at it in multiple ways, but right now I'm just showing one type of DNA methylation, line 1. Those children that live with higher neighborhood poverty around them are the ones that are showing increased line 1 methylation. So we're seeing a relationship of adversity on DNA methylation. This is, I just did for this presentation, I did a quick and dirty analysis without controlling for anything, and I'm seeing a borderline relationship of the DAPs during pregnancy, the OP metabolites during pregnancy, and DNA methylation. So we're seeing social adversity affecting DNA methylation and exposure to DAPs, or organophosphates related to DNA methylation. The next step is looking at how do they synergize, interact with each other. As we go forward, again, we're going to need to have large cohorts to be able to look at these complex interactions. There's something called the PACE Consortium that compiles cohorts from around the world looking at DNA methylation and different factors. And the reason why this is important for the 21st century is we're seeing an uptick worldwide in the number of conflict and violence in the world. And so we can't look at chemical exposure alone. We need to look at their interaction with social adversity to look at the effects on children's health. So that's my next point in the 21st century. So we need to look at the neurotoxic exposome, the whole mixture. We need to take into account the genetics and the neurobiology, and we need to consider the stress factors. And we're particularly interested in how this all comes together to affect the criminality of the children. So in summary, environmental factors affecting our children in the 21st century are going to be the unstudied, unidentified, regrettable substitutes. They're going to be the creation of the complex chemical mixtures and their potential synergies. And there's going to be an increase in social stressors and interaction with chemical factors. But in case you're not already depressed, since I'm interested in social adversity, probably the greatest concern we have for the 21st century is climate change. It is the biggest environmental emergency that's going to affect our children. So it's estimated that by 2050, 1.45 billion children will live in areas where the temperature will rise by 2 degrees centigrade, and by 2100, 74% of the world's population will have 20-plus days of lethal heat per year. There's going to be increasing conflict with limited resources. I already see that in the places I work in Africa. And there's going to be, as we already see, environmental refugees. There are not that many studies, surprisingly, on the effect of climate change, primary research studies on the effect of climate change on children's health and on pregnancy. But these studies are already showing that there is an increase in the odds of stillbirth associated with high heat days. Something happened to this slide. With temperature increase and precipitation decrease, there's been association with lowered birth weight. There's also, in terms of the children themselves, that there's been an increase in immediate pediatric hospital admissions soon after a temperature increase, and this is in Vietnam. There have been studies in California by Nino Kinsley and others that look at an increase in respiratory problems, in health effects associated with California wildfires, which is related to climate change. And of course, there's an increase in infectious disease that has been noted here in Taiwan in relationship to torrential rainfall, which is also part of climate change. And you can see across the bottom there, what's going to happen to bring this all back together is that with increases in infectious disease, there's going to be increase in use of pesticides, and there's going to be increase of hazardous waste. So here's a study that was done here in China, that there was a concomitant increase in pesticide use with increase in precipitation and with temperature. In our study in Africa, I've done a birth cohort study in Africa, we are particularly concerned because we're already seeing last year an amazingly horrible outbreak of malaria in the community that I work in. And with that community, we also see that there's going to be an uptick in the use of pyrethroids and DDT because of the increase in malaria outbreaks. There's also going to be an increase with climate change in the number of children who are undernourished at under age 5. So all of these things of climate change are going to affect those that are already at high risk. So I'm asking all of us to be the generation of change, to hand over hand over a better earth to the next generation. And I know this is very ethnocentric of me, but we developed this little brochure that for the United States, for California, but I'm asking all of you to consider thinking about in your own communities, what can you do to prevent or to stop the pace of climate change? We as individuals need to do something, as well as policymakers need to do something. And I recognize that I'm coming from the country that's doing probably one of the least right now, but I live in a state that's doing one of the most. So, so I just want to point out that this is not going to work in Taiwan, and this little brochure is not going to work in whatever country you come from. But why don't you develop something like this for the populations that you work with to be able to change the communities that you work with? These are kids that are working on our studies, and they're from high schools around the Bay Area that we have motivated to work on climate change issues. So with that, I just want to point out that children are 1/3 of our population and all of our future. And there's no plan B. We only have one Earth, and let's do what we can to keep this Earth alive. I'd like to thank my collaborators, in particular Kim Harley, Asa Bradman, and Nina Holland. And I'd like to have a special thanks for the families that are working in CHAMACOS. I've followed them for so long, I feel like they're my children. And also to our funders. And thank you.
And your methods is really amazing, the cluster approach. I think it's wonderful. I'm have to say sorry to you because of the time. We have to skip the question, but please, Dr. Kim has something for you.